All right, now we're talking about Chapter 13, Nervous System, Brain, and Cranial Nerves for Anatomy and Physiology, Bio 246. And the objectives which you should be able to fulfill after this section should be to describe the general regions of the brain. We're talking about the brain organization and development. Describe the process of nervous tissue development and neurulation. Provide the scientific names for the embryonic forebrain, midbrain, and hindbrain. Name the 5 secondary brain vesicles. Describe their embryonic origins and list the adult brain structures that are formed by each. Compare and contrast the general composition, function, and distribution of gray and white matter throughout the central nervous system. And with the brain regions, an overview of the basic brain anatomy, we have our cerebrum, which has 2 hemispheres, 5 lobes per hemisphere, so 10 total. We have a diencephalon, or diencephalon, and we have the brainstem and the cerebellum. The outer surface is folded. We have our gyri, which are basically ridges, plural of gyrus, and sulci, which is the plural of sulcus, depressions between those ridges. The fissures are deep sulci. We have the anterior, which is the rostral, and the posterior, which is called the caudal. So with the human brain, we'll take a closer look here. And we're going to have a lot of location that's going to be vital for us with the brain that you're going to need to know for both lab and for the class. So look at the brain here, looking for the anterior. We have our frontal lobe here in the anterior. And we have a gyri. Every time you see one of these guys here, these are gyri, or that's a gyrus. And the sulcus is the space between them, or sulci for plural. So here we have our lateral sulcus here, separating the temporal lobe from the frontal lobe, and the parietal lobe and the occipital lobe. We have all these little sulci that are between here, or sulcus for a singular at a given specific area. We have our central sulcus here, the posterior, the parietal lobe, the parietal occipital sulcus, the occipital lobe, and below we have here in the brain stem, we have the pons, the medulla oblongata, and these are cranial nerves, little projections. You're seeing parts that are cut off here. Cranial nerves are coming to play a little later for us here, and there's a cerebellum, and of course our spinal cord. So for the inferior view, the anterior with our eyes here, and then we have frontal lobe here, temporal lobe, the side there, occipital lobe, and the posterior here, the caudal region, the cerebrum. And then we have here the olfactory bulb, the olfactory tracts. Tracts will come into play, talking about a little more later on. The optic chiasm. We have our optic nerve, pituitary gland, which obviously is an important gland for us, the optic tract, the mammillary bodies, which is part of the diencephalon here, or diencephalon, the midbrain here, and we have our pons, medulla oblongata, all part of the brainstem, the midbrain, the pons, the medulla oblongata, brainstem. We have, again, more cranial nerves here. You will have to know the 12 different cranial nerves and the cerebellum. And then from our mid-sagittal view, you have to go over these as many times as you need to until you get them, and we'll have worksheets for them as well. So here we have, again, the anterior portion of the mid-sagittal view, the cut right down the middle there, so to speak. We have our frontal lobe, our septum pellucidum, our corpus callosum here, and then we have part of our diencephalon here. We have the epithalamus right in here, and then the pineal gland, and we have our thalamus, our intrathalamic adhesion, okay? We have our hypothalamus, our mammillary body. We have the pituitary gland, and then going down here, the temporal lobe, and then we have the brain stem here, for midbrain. We have our pons, medulla oblongata, and the spinal cord. Then more the posterior, our central sulcus here, our parietal lobe, our parietal occipital sulcus, our occipital lobe, our tectal plate, our cerebral aqueduct, space between there, fourth ventricle, and ventricle is a space where places things are going to move, right? And of course our cerebellum once again here. Now, kind of as before, any area that we haven't read, it's not that it's not important, it's that we're not going to spend a lot of time on for your need to know right now. Down the road, of course, you should understand the application. We have to understand the basics before we get to the application. But yes, this is ultimately vital, just not in the moment. With traumatic brain injury, it's an acute brain damage occurring as a result of trauma. Concussions are the most common type of traumatic brain injury. There's temporal loss of consciousness with headaches, drowsiness, confusion, and amnesia is possible. It can have cumulative effects on intellect, personality, and mood. long-term, not just short-term. Contusion is basic bruising of brain due to trauma. Contusion in general through the body is the same thing, just anywhere, but specifically a brain contusion here. Second impact syndrome is a second injury before the first resolves, which develops severe brain swelling. You've already got damage and you have another impact. Not a good thing, right? Progesterone is possibly therapeutic for traumatic brain injuries. And as far as development of brain divisions, neurulation is another thing I'm going to cover very quickly here. You're not going to have to know for the moment, but it will come into play in later units, probably more so in second semester for content. That said, neurulation begins in the third week of embryonic development. It's part of the embryo's ectoderm layer. It's the thick neural plate. The notochord beneath it induces the neural plate to form the neural tube, which develops into the CNS, central nervous system. The neural plate develops the neural groove, and cells proliferate along these lateral plate margins. They become neural folds, and their tips become neural crests. I'm not going to go into great detail with that for the moment. Just go into the basic steps here. Neural groove deepens as the folds rise and they approach one another, and the crest cells separate and form neural folds. And by the end of your third week, these folds meet and form the neural tube and surrounding neural canal for the embryo. And the fusion begins in the middle, then progresses superiorly and inferiorly, and the openings of the cranial and caudal ends of the tube, the neural pores close by the end of the fourth week. As far as development of brain divisions, for a short time, the neural tube opens at both ends, the cranial neural pore and the caudal neural pore. If openings do not close, the neural tube defect will occur with anencephaly and spina bifida, which are areas that we're going to cover very briefly here. Again, more content for a later time, but it's good to know generally for the short term here. Serious developmental deformities of brain, the spinal cord, and meninges is occurring with such neural tube defects. The risk is lowered by taking vitamin B12 and folate in pregnancy, and really true in general from a standpoint of other problems, but yes, it's specifically now. With anencephaly, it's substantial or complete absence of a brain. The infant, of course, dies soon after birth in this instance. Spina bifida, there's a failure to close the caudal portion of the neural tube. Spina bifida cystica is there's almost no force in the vertebral arch, and there's a large cyst in the back. This often causes paralysis of lower limbs. Spina bifida occulta is a partial defect of the bony arch. It's less serious, but it's a lot more common. As far as the primary brain vesicles, those form by the fourth week of the cranial from the cranial neural tube. The forebrain, which is the procephalencephalon. It's a future cerebrum, essentially here. Okay, we're talking about here. It's eventually become the cerebrum, and it's which includes the telencephalon and the diencephalon. The midbrain, which is the mesencephalon here, is basically regulates the auditory and the visual, and we'll mention that more again here shortly. And the hindbrain, it's kind of nice to see things all at once, which we're talking about the rhombencephalon. We're basically talking about the future cerebellum, the pons, and the medulla oblongata. So by the fifth week, the telencephalon becomes the cerebrum. Okay, it's going to become your cerebrum. And the diencephalon becomes the thalamus, hypothalamus, and epithalamus, right here. The mesencephalon becomes the midbrain. The metencephalon becomes the pons and the cerebellum. And then the myencephalon becomes the medulla oblongata. In 13 to 26 weeks, the brain continues to develop in the fetus. The telencephalon envelops in the diencephalon and develops in the surface folds. And these folds allow the brain to fit within the cranial cavity. And the most gyri and sulci are present by birth. Now, as far as the gray and white matter distribution, the brain and the spinal cord is composed of the gray and the white tissue. It's often confused with your brain capacity, and whether you're using your full brain for your full intellectual capacity, when they're confusing the fact that white is more your wiring, where your CPU is your gray matter. So you're not going to be using your white matter for storage of memory or for integration of information. It's more connections between the gray matter that's doing that. So you couldn't possibly use all of your brain from that standpoint because it's not technically all a CPU. The gray matter is the CPU, the central processing unit, like a computer. It's made of neuron cell bodies, dendrites, and unmyelinated axons. The cortex is the superficial layer of the gray matter. Nuclei are regions of gray matter, clusters of cell bodies that are either close to the surface or deep within the brain. The white matter is your wiring. It consists of the myelinated axons. It's organized in bundles called tracts. And again, we'll talk more about tracts at a later time. So here we have all of our gray matter, right? See all the gray areas. And we have the white matter, typically more deeper here. The cerebral cortex, part of the gray matter, cerebral nuclei throughout, okay? And then the white matter is the corpus callosum and the internal capsule as examples here, primarily for that white matter in the diencephalon and the cerebrum. As far as the cerebellum and the brainstem here, I don't think we have to zoom in for this one here necessarily, but seeing this here, you can see the cerebral cortex, and you have the inner white matter, of course, and we have our outer white matter as well, and we have our inner gray matter, these little areas here for the brainstem. And by seeing it, this being a picture more here, showing here the cerebellum, and of course the cerebrum being the front here for A, and then we'll talk more about the rest here in a short bit here. Actually, I'll come back to that. Because the medulla oblongata here, right, basically showing here and cutting that cut here, going to get a nice little cut there, basically horizontal or transverse plane type cut. We have our inner gray matter, and we have our outer white matter. And then we have for the spinal cord, actually it's flipped in the spinal cord versus typically for the brain, for the white matter being more external and more internal is more the inner gray matter, including the medulla oblongata. So, what did you learn? What are the 4 major regions of the brain? How does the neural plate form a neural tube? Identify the 5 secondary vesicles and list the adult brain structures they form. And where is gray matter located within the cerebrum and the spinal cord? And that's section 13.1. We'll come back for section 13.2 here shortly.